Okay, everybody, Chapter 6, Amino Acid Metabolism. So our lecture objectives, we're going to know and discuss the biomedical importance of amino acid metabolism in relation to various amino acid deficiency states causing diseases. Describe the importance of nutritionally essential and non-essential amino acid requirements in humans. Demonstrate biosynthesis of amino acids as grouped by metabolic precursors. We've seen that a little bit in Biochem 1 and in previous chapters here. Describe the role of cofactors, particularly, it's a one word I can't say, particularly pyridoxal phosphate, tetrahydrofolate, and tetrahydrobiopterine in amino acid metabolism. I can say all the fun words, but not particularly. Okay. Demonstrate the amino acid degradative pathways. Identify and know the glucogenic and ketogenic amino acids. Explain mechanisms of defective amino acid metabolism leading to certain diseases in terms of defective enzymatic activities. Okay. So, amino acids are derived from the diet or from the tissue proteins. So either we're consuming them or our body's going to break down proteins in order to get them. Amino acids cannot be stored in human tissues. Okay, so we cannot store them as amino acids. Amino acids are either recycled and incorporated to synthesize peptides and protein, or are catabolized by oxidative degradation to supply significant amounts of metabolic energy, or converted to another amino acid or to some other metabolically important biomolecules. So real quick, let's break those down. So all of our amino acids, they're either going to be recycled, so they're going to be reused and used to make proteins. So making a peptide chains turn into proteins. So amino acids, so fate of amino acids. We have make proteins, recycle them. We're going to catabolize by oxidative degradation. So if you remember catabolism, catabolic specifically, talking about the breakdown of complex molecules such as proteins or lipids. So we can break them down and turn them into energy. That's that metabolic energy part. Or converted to another amino acid or to some other metabolically important biomolecule. So change to another amino acid or something else that we need. Again, all of these kind of involve recycling and reusing the amino acids to make sure that nothing goes to waste. So several proteases. Remember, protease is going to be an enzyme that breaks down proteins. And peptidases, again, enzyme that breaks down peptides, catalyze the degradation of proteins and peptides to their constituent amino acids. So biomedical importance. We're going to go over some of these conditions. So these conditions will come up several times, as well as I guarantee you'll have at least one of these pop up on your part 1 boards, or something very similar to them, or there'll be an answer choice and you want to make sure that you know what it is so you don't accidentally pick it. Okay, amino acid deficiency states results. What's worded a little bit weird. So the amino acid deficiency results due to either poor sources of any of the nutritionally essential amino acids. If you remember the essential amino acids, Private Tim Hall, here's a good chance for you to review those if you can remember what they all are. If not, take a second, go back, look through those again right now, because I promise you you're going to need to remember these because you're going to have questions on them again. So, nutritionally essential amino acids from the diet or present in inadequate amounts. For example, certain grains are relatively poor sources of tryptophan and lysine. So a good source of total protein, such as milk, egg, fish, or meat, should be supplied to avoid deficiency of these amino acids and protein deficiency conditions. So in a lot of poverty-stricken regions, where, let's say in Asia or Africa, Middle East, where they might only have specific grains, rice or wheat, and not a lot of the milk, the egg, the fish, meat, for consumption, especially in the lowest classes of people, they can end up with these protein deficiencies because of not consuming appropriate amounts of our animal products that have these proteins. This is also one of the reasons why when somebody decides to be vegetarian or live a vegan lifestyle, diet is incredibly important. That they monitor their diet and they get those higher quality plant proteins, the quinoa, like the quote unquote superfoods, that have more of the protein sources, as well as supplementation often is necessary for those kind of lifestyles, with the foods that we have available. So looking at diseases, there's a key difference between these two, and you will be asked this as a test question to differentiate between these two. There's kwashiorkor, which is a protein deficiency. So this is somebody, they could be eating 3,000 calories a day, but of just ramen noodles or just rice or something like that. So this is not a calorie deficiency, just a protein deficiency. So in some regions of the world, children are weaned onto starchy diet poor in proteins, and that leads to the kwashiorkor. Marasmus, this is both protein and calorie deficit. So an insufficient caloric intake and deficiency in some specific nutritionally essential amino acid intake can lead to marasmus. So if we look, and there is a little bit of a difference between these two. So this is kind of what you expect to see with kwashiorkor. So you see that the extended belly, very thin in the extremities. That extended belly like that is very characteristic of kwashiorkor disease. It's actually spelled wrong here. Kwashiorkor. Now, marasmus, this is just very much that you can see this marasmus versus kwashiorkor. So that extended belly with kwashiorkor, marasmus, just almost skin and bones, is really what that sort of looks like. And that's the big difference with the calorie deficit, the calorie deficiency that they have with marasmus. So those are the two diseases that we talk about right now. So amino acid metabolism includes biosynthesis of amino acids, as well as degradation or breakdown of amino acids. Of the 20 common L-amino acids that are used in humans to synthesize proteins, 11 are synthesized by the human body, while the other 9 are nutritionally essential amino acids and must be obtained from dietary sources. Here's our Private Tim Hall: phenylalanine, valine, tryptophan, threonine, isoleucine, trying to do these in the correct order, methionine, histidine, and also technically arginine. Arginine is that semi-essential one. whereas in children is synthesized at rates inadequate to support the growth of a child. So we need arginine to be produced as well. And then our leucine and our lysine. Yeah, so arginine. Even better, I would just take that out. Just say arginine is essential. So although arginine is produced in the urea cycle, it is immediately degraded to produce urea, so we need it from the diet. So 11 nutritionally non-essential amino acids are synthesized by the body by the common intermediates. So from glucose or intermediates of the glycolysis or the TCA cycle. So we think a lot of our oxaloacetate or malate or fumarate or citrate, all of those guys can be converted back into amino acids. Also from another amino acid, we can add or take away in order to get the correct amino acid that we need. Also from ammonia, so ammonium ion, the NH4, kind of that base that we see, it's in part of the structure of our amino acids. So the 11 non-essential amino acids are alanine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, hydroxyproline, proline, serine, tyrosine, and it has arginine on here again, but again, like, we count it more as one of our essential amino acids. Tyrosine and cysteine are sometimes termed as semi-essential because mammalian tissues can synthesize these two amino acids when the two essential amino acids, phenylalanine and methionine, are available. Remember, we talk about essential. Essential meaning that we have to have them in our diet. Non-essential means our body can produce them themselves. So there are three major sources of amino acids to maintain the pool. We have digestion of endogenous proteins and peptides. So these are our digestive enzymes, bile, intestinal mucus, our dead intestinal epithelial cells. All of the stuff, like the proteins that get broken. Remember, we want to recycle a bunch of our proteins. So anything that gets kind of released into the stomach, or into the stomach or into the intestines, we're going to break down and reabsorb a good chunk of that. So about 85% of amino acids result from endogenous protein digestion. 85% of them that go through get reabsorbed. The remaining just get excreted in the feces. We also have dietary protein after digestion and absorption of the resulting amino acids. It's about 100 grams per day. And also intracellular protein turnover or degradation is 230 grams per day. So this is like our big hitter. This is mostly what we're doing. A lot of turnover, breaking things down and rebuilding is what we're going to see the most to get our protein needs. To get our amino acid needs, I should say. Specific amino acid transporter systems are present for the release and uptake by cells. Most aliphatic amino acids aliphatic means relating to or denoting organic compounds in which carbon atoms form open chains, as in alkanes, so not aromatic rings, so chain structures. The chain structures are our aliphatic amino acids. So most of our open chain amino acids are transported by a sodium ion dependent and sodium ion independent transporters. For the transportation of acidic, basic, amino acids and glutamine, more restricted type of transporter system exists. Some amino acids are also subject to competitive release or uptake by cells by the same type of transporter system. Just clicked the wrong thing. We're going to have something open up here in a sec. Let me close that. Sorry about that. Okay. Alanine, aspartate, and glutamate are formed by one-step transamination reactions. Think back to Biochem 1, transamination. Kind of what is that sort of reaction? What does that mean? A transamination, so it is a transfer of an amino group from one molecule to another, especially from amino acid to a keto acid. So amino acid plus pyruvate, we get alpha keto A and alanine, amino acid plus oxaloacetate, and alpha keto A and an aspartate, any amino acid plus the alpha keto glutarate, alpha keto A plus glutamate. And also alpha keto acids, alpha keto acid right here, are the deaminated form of amino acids. So that amine group, we're removing that amine group off of them. So these have been used in several applications in medicine, food and feed industries. In vivo, alpha keto acids are ketogenic or glucogenic precursors, which regulate protein turnover. When we talk about ketogenic and glucogenic, keto referring to the ketone bodies. So those are ketogenic, they can be turned into ketones, versus glucogenic, which can be converted, the amino acids, which can be converted back to glucose. That's what ketogenic and glucogenic is referring to. So here is a simplified outline of amino acid biosynthesis grouped by metabolic precursors. Okay. So from glucose, we have glucose 6. Glucose goes to glucose 6-phosphate. Remember the enzyme that's here. We can have hexokinase or glucokinase. Hexokinase is in the muscles. Glucokinase is in the liver tissue. And we have the different steps where we convert it to ribose 5-phosphate, which we can then get histidine from there. Or our glucose 6-phosphate can go through the steps of glycolysis to give us the 3-phosphoglycerate, and from there it can be converted to serine, or we can convert it to the erythrose 4-phosphate, which goes to tryptophan, phenylalanine, and tyrosine. Can continue to phosphoenol pyruvate. Serine can also be converted to glycine and cysteine. Phosphoenol pyruvate then goes to pyruvate. Pyruvate can be converted to alanine, valine, leucine, and isoleucine. Pyruvate eventually goes to citrate in the Krebs cycle, and alpha ketoglutarate and oxaloacetate, which then come down into all of these other amino acids. So this is really kind of the simplified version of it. I would highly, highly, highly recommend that start this early. Do not fall behind in this chunk, because you're going to have a handful of questions that are just quite simply, which of the following turns into this? Which of the following turns into that? Through what process does this turn into that? Granted, it's going to be a little bit wordier than that typically, but that's the overall idea. As additionally, you will see some of this stuff on boards. What I would recommend doing is drawing this out yourself several times, and then coming up with little mnemonics or something to help you remember these things. So if we also look at this, tryptophan, phenylalanine, and tyrosine, your pal Tyrone trips you in the ring. These are all ring structures. So from our phosphoenol pyruvate, our erythrose 4-phosphate, these are our ring structures. Our alanine, valine, leucine, and isoleucine. Think kind of like evil, but avil. I don't know. Any sort of way that you can try and remember which ones are grouped where will help you to start to develop those chains. And I would highly recommend draw this out once a day at least for a week. Take 10-15 minutes out of each day, draw it once a day. The next week, every other day, then draw it once or twice a week, and all the way up to your exam. And as soon as your exam starts, if you're allowed a scratch paper, immediately jot this down. Again, ask your teacher. If your teacher doesn't allow you a scratch paper, I know with certain testing systems, depending on whether you're in person or outside of or however, talk to your teacher and ask and make the request to be able to use scratch paper. It's definitely very beneficial in my opinion, for taking exams. Okay. Continuing on, so alpha-ketoglutarate is the precursor of glutamate, glutamine, proline, and arginine. Glutamate is produced by transamination reaction with the alpha-ketoglutarate, catalyzed by the enzyme amino transferase, also known as transaminase, requiring pyridoxal phosphate as a cofactor. So if we start to remember pyridoxal phosphate, we've definitely heard that one before. It's going to be one of our B vitamins. Remember our B vitamins? Tall, rich, nudists played pickleball for centuries. See if we can remember all of our different B vitamins. Remember, it goes B. Let me make sure. Yeah, we go 1, 2, 3, 5, 6, 7, 9, and 12. Thymine, riboflavin, niacin, pantothenic acid, then our pyridoxine or pyridoxal 5-phosphate is the active form, biotin, folate, and cobalamin. Those are our B vitamins. So it's 1, 2, 3, 5, 6, 7, 9, and 12. So we're looking at B6 is our pyridoxal phosphate. And glutamate can continue to be broken down to produce glutamine, proline, and arginine. Glutamate produces glutamine by amidation reaction catalyzed by glutamine synthetase. I would even start within this. I would start adding these in. So here, this is amino transferase, aka transaminase, with B6, and then here amidation reaction, glutamine synthetase. Start adding these in here as well. Again, the more that you can do repetitions in all of this, the better off you're going to be. And it's gonna kind of suck, honestly, like, to have to do this over and over again. But if you start early, you'll be in a much better position. Because I promise you, these are the sort of questions he's gonna ask. A handful of these questions, and if you start it now, you'll be well prepared by the time you get to the next test. 3-phosphoglycerate is the precursor right here. 3-phosphoglycerate is the precursor to serine, which can then be converted to cysteine and glycine. Glycine can also be formed from CO2, NH4, and in N5N10 methylene tetrahydrofolate. If we're thinking tetrahydrofolate, methylene tetrahydrofolate, especially tetrahydrofolate is our B9. It's our active form of our B9. And the reaction is catalyzed by glycine synthase, all these enzymes we want to remember, in the presence of tetrahydrofolate and pyridoxal phosphate as cofactors, as well as write out those cofactors. You can also do this whole thing, set this whole thing up as a graph or chart as well, and if that helps you to study. The sulfhydryl group, SH of cysteine, is donated by methionine. Oxaloacetate produces aspartate. Going back to our pentose phosphate pathway. If you don't remember our pentose phosphate pathway, I'll pull up see if I can find a good, simpler. This was our pentose phosphate pathway. Glucose 6-phosphate to the 6-phosphoglucono delta lactone, phos-6-phosphogluconate, and then our ribulose 5-phosphate, which that was then you could be used in our nucleic acids or converted back into glucose. One of the ending products of that we can have is erythrose 4-phosphate. Gives us to the tryptophan and phenylalanine and tyrosine. This is sorting it. This whole chart is kind of sorting into just more of the metabolites. So this is from glycolysis, from the pentose phosphate pathway, or from the citric acid cycle, breaking it down. Another chart just breaking it down, similar to this. Whichever is the easier way to do it for you. Aspartate is converted to asparagines by amidation reaction. Asparagine synthase. Aspartate also generates methionine, lysine, threonine. Threonine is converted to isoleucine. So you can see a lot of this chapter is just sort of listing off what can go to what. That's what's going to take up your most time in the chapter. And please take the time to do it. Do it now. Start early so that you don't have to struggle to try to memorize all of this later on. Okay? Let it be a continuous process rather than a night before the exam. So then threonine is converted to isoleucine. Pyruvate produces alanine, valine, leucine, and isoleucine. Phosphoenol pyruvate and erythrose 4-phosphate produce phenylalanine, tyrosine, and tryptophan. And the ribose 5-phosphate produces histidine. That's all of our different conversions of an amino acid or intermediates going to different amino acids. Just straight up memorize them. There's not a lot else you can do. I'm sorry. Okay, we're talking about the role of certain cofactors in amino acid metabolism and their brief mechanisms of action. So amino acid metabolism necessitates the involvement of 3 important cofactors as described below. So we're going to focus on 3 of them: our pyridoxal phosphate, our tetrahydrofolate, and our tetrahydrobiopterin. So the pyridoxal phosphate, it is a derivative of pyridoxine, also known as B6. So pyridoxine is the name of B6, and the active form is the pyridoxal phosphate. It is required in transamination reaction as a cofactor and involved in the transfer of the amino group of the amino acid to an alpha keto acid. So it is the amino acid plus alpha ketoglutarate to the alpha keto acid and glutamate. So this little phrase down here, that's what you really want to know. Tetrahydrofolate, aka B9, that ending put the folate should give you the clue. Folate is our B9. Tetrahydrofolate is its active form. And this coenzyme is derived from the vitamin folic acid. This participates as a cofactor to accept or donate a 1 carbon group, also known as, most commonly, methyl transfers. Methyl transfers is the big one for that. And actually, let me see if I can find the... there should be a picture that I have. Here we go, the methylation cycle, where we have a methionine and homocysteine kind of cycle, and B9 and our B12 are both involved in this methyl transfer in order to maintain the levels of homocysteine and stuff throughout our body. This especially, I won't get too deep into this, but this is a really important reaction for maintaining homeostasis within the body. So our folate and our cobalamin are both involved in that. So for example, so we donate 1 carbon unit group, and that most commonly is our methylation. So serine is converted to glycine. 1 carbon group is donated from serine to tetrahydrofolate. This reaction requires presence of THF and pyridoxal phosphate, and serine gets converted to glycine. Glycine being our smallest and our simplest amino acid. Tetrahydrobiopterin, BH4, this coenzyme is required for aromatic ring hydroxylation. So aromatic ring. The reaction involves molecular oxygen, and 1 atom of oxygen is incorporated into the product. The second oxygen atom is incorporated in water. So phenylalanine produces tyrosine is the example of that. Now, overview the synthesis of the non-essential amino acids. Again, another chart. Okay, so I'm saying a lot of these. If you can find a way to organize them all, that's really what you need to do, and just practice and put all of these different charts, because most of them, they're saying very similar things. If you can put them into one massive chart and one space and organize it that way, that's probably going to be your best bet. Degradation or breakdown of amino acids. So the carbon skeleton of the 20 standard amino acids can be entirely oxidized to carbon dioxide and water. The carbon skeleton of these amino acids can also be used to synthesize glucose or ketone bodies and fatty acids. So glucogenic amino acids are degraded to pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate, and can be used as the precursor of glucose. Ketogenic amino acids, let me actually... There is a little mnemonic to remember the ketogenic. Lucy lies to get the keys. Our leucine and lysine are our ketogenic amino acids. The glucogenic amino acids, there are actually 13 different glucogenic amino acids. So any amino acids that are coming from or being broken down, if you can form an amino acid from pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, or oxaloacetate, those are going to be our glucogenic amino acids. So if we come back up here, glutamate, glutamine, proline, arginine, aspartate, asparagine, methionine, threonine, lysine, alanine, valine, leucine, isoleucine, these guys, all of these guys here are going to be also glucogenic. I'm not super clear if there's an overlap between those. So our okay, so this is what it says. It's a little bit different, not quite as clear. But leucine and lysine are exclusively ketogenic. The 5 more are both ketogenic and glucogenic. Phenylalanine, isoleucine, threonine, tryptophan, and tyrosine. And then the remaining 13 are exclusively glucogenic. So depending on how you word the question, the exclusively glucogenic is the big thing. So that's how that works. Okay. The amino acids can be grouped according to their common breakdown products. Alanine, cysteine, glycine, serine, and threonine are degraded to pyruvate. Asparagine and aspartate are degraded to oxaloacetates. Arginine, glutamate, glutamine, histidine, and proline are degraded to alpha-ketoglutarate. Little mnemonic for this: GGs are how players get killed. Isoleucine, methionine, and valine are degraded to succinyl-CoA. Ideal men value co-sleeping. I don't remember most of these. I did these at some point. Threonine, isoleucine, leucine, and lysine are degraded to acetoacetate and acetyl-CoA. That is thrall. Tryptophan is degraded to alanine and acetoacetate. Phenylalanine and tyrosine are degraded to fumarate and acetoacetate. There is a chart. I'm trying to remember what the chart is from Biochem 1, but there's a whole... It was this one, actually. This chart. This is the chart. We looked at this a little bit in Biochem 1. Kind of how the malades, glucose, how it goes to different things. So we introduced this last try. We didn't do a ton with it, but now this try, we're really digging into it. Here's a degradation of ketogenic amino acids. The exclusively ketogenic acids are leucine and lysine, and then another 5. So there's 7 here total. Leucine and lysine are exclusively ketogenic, but then a threonine, isoleucine, tryptophan, phenylalanine, and tyrosine are all both ketogenic and glucogenic in nature. Here we go. These are the ones that are both ketogenic and glucogenic. The strictly ketogenic are lysine and leucine. And we have a little clinical nugget here. So defects in amino acid metabolism may lead to several diseases. You will have a couple of questions covering these diseases, so good thing to take note and try to remember these as well. So defects in glycine metabolism causes excessive oxalate production, which can lead to one class of kidney stone formation and renal failure. This will come back not super soon, but as you get deeper into the program, these will come back. You'll talk a lot about renal failure and renal diseases in Phys 2 next trimester, as well as in Touch and Phys Tri 4, and a decent amount in systems pathology as well in Tri 4. Tri 4. So the defective enzyme in this is the glycine transaminase. The product that accumulates is glyoxylate. This disease is known as primary oxaluria type 1, and the symptoms are renal failure due to stone formation. We're going to find a picture of that. See the stone formation. Nephrolithiasis as well is what you'll hear it be called. Okay. Next condition, defective methionine degradation can cause hyperhomocystinemia, which is linked to blood clotting disorders and heart disease. The defective enzyme is cystathionine beta synthase. The products accumulated are homocysteine and methionine. Disease is known as homocystinemia. So this is actually what I showed you just before. Where'd it go? This reaction right here between the methionine and the homocysteine, that's what this condition is talking about. That the defective enzymes cystathionine beta synthase, and you get this homocystinemia, you get cardiovascular complications and neurologic problems. And then we also have defective phenylalanine degradation can lead to phenylketonuria or PKU. And albinism. It's PKU. I think PKU, if I'm not mistaken, is more common in albinism. Yeah, so albinism is actually an effect the albinism is caused by the disease PKU. I'm trying to remember, I believe PKU management is literally this. It's a lifetime diet with very limited intake of foods with phenylalanine. So essentially, your body can't process the phenylalanine, so you literally just have to avoid it for your entire life, which again it's just a very strict dietary condition. This is something that typically they'll see the albinism, they'll catch this early because of the presentation, as well as like if they'll start to get sick really quickly after starting any food, even like breast milk, because it's going to have the amino acids in it and they can't break it down. Here are more of those conditions. Let's see. This is going to go down and give you the names of the conditions and kind of their symptomatology. I'm not going to go over all of these. So there's a lot of information in here as well. I was best thing to do here, I would honestly say break some of these up, separate these out, just because right here it looks really, really busy. We'll go over a couple of these. So alkaptonuria, aka black urine disease, is a very rare inherited disorder that prevents the body from fully breaking down two protein building blocks called tyrosine and phenylalanine. So you lose the ability to break down the tyrosine and the phenylalanine, and you get black urine disease. Here's our PKU, where we can't break down the phenylalanine. Tyrosinemia, you see emia, it's blood related, is a genetic disorder characterized by problems breaking down the amino acid tyrosine, which is a building block of most proteins. If the condition is untreated, tyrosine and its byproducts build up in tissues and organs, which can lead to serious health problems. There's several different types: tyrosinemia 1, 2, and 3. Okay. These are where test questions will come from. Knowing the different enzymes and kind of what they cause or what the name of the disease is. Where the defective enzyme is phenylalanine hydroxylase, where the defective enzyme cannot convert phenylalanine to tyrosine. And actually, let me see. One of the reasons why tyrosine and why this is so important, phenylalanine to tyrosine. Tyrosine is used to create dopamine, norepinephrine, and epinephrine, as well as melanin. So this is really, as well as T3 and T4, there are thyroid hormones. You should be learning about those more about those in next trimester in Phys 2. But tyrosine is incredibly important, where we need it for our dopamine, norepinephrine, epinephrine. If you remember from Phys 1, we should have learned a little bit about these, where our norepinephrine and epinephrine are both hormones as well as neurotransmitters. And dopamine is incredibly important within the brain for regulating emotion, regulating movement, a whole bunch of stuff with dopamine. Dopamine deficiency is actually one of the things that leads to Parkinson's. You get damage to the substantia nigra, it's called, in the brain. Dopamine is affected, and you have Parkinson's disease. So the high levels of phenylalanine and phenylpyruvate, or phenylketone, in the blood or urine are accumulated. You'll get neonatal vomiting and mental retardation. Again, because you can't produce the things that you need for your central nervous system, for your brain. Defective phenylalanine and tyrosine degradation can also lead to alkaptonuria or alkaptonuria. It's just spelled different with a K or with a C. Same thing, though. The defective enzyme causing alkaptonuria is homogentisate oxidase, also known as homogentisate 1,2-dioxygenase. It causes accumulation of homogentisate oxidation, of which causes dark pigmentation, black urine. You get dark pigmentation of the urine and late developing degenerative arthritis. Now, defective branched-chain amino acid metabolism. Branched-chain amino acids, valine, leucine, and isoleucine, remember the mnemonic for our branch chain, live on a branch. These are oxidized to produce corresponding alpha keto acids. Conversion of these amino acids to their corresponding alpha keto acid is catalyzed by branch-chain aminotransferase, especially within muscle, adipose, kidney, and brain tissue. These branch-chain alpha keto acids are further oxidatively decarboxylated to form corresponding acyl-CoA derivatives. This reaction is catalyzed by branch-chain alpha keto acid dehydrogenase complex. Real big mouthful. That whole guy. So something's going to be wrong with this guy. And that causes what's called maple syrup urine disease. This is a rare genetic disease where the 3 branch-chain alpha keto acids, as well as their precursors, the branch-chain amino acids, the valine, leucine, and isoleucine, accumulate in the blood and then are distributed in the urine. So they are filtered out into the urine. The products that accumulate appear in urine and produces the smell of maple syrup imparted by alpha keto acids. Kind of like very similar to somebody who is diabetic going through ketogenesis, or even if you have somebody who's doing a very, very strict keto diet. A lot of times if you smell their breath, it'll smell sweet and fruity. So same thing is happening here, where because you have the alpha keto acids, they produce that sweeter smell. Some of the symptoms, you can have vomiting, convulsions, abnormal development of the brain, mental retardation, poor myelination, and early death. A defect in proline metabolism can cause hyperprolinemia type 1. It is characterized by defective function or inhibition of proline dehydrogenase. Well, that's the enzyme we'll want to remember. Proline dehydrogenase is normally responsible for the conversion of proline to form glutamate semialdehyde, followed by its conversion to glutamate and finally to alpha ketoglutarate. So we can't get from proline to alpha ketoglutarate. Hyperprolinemia type 2, in this disease the defective enzyme is the glutamate semaldehyde dehydrogenase. So this is the hyperprolinemia type 1. The first enzyme is inhibited. Hyperprolinemia type 2, the second enzyme is inhibited. The defect in proline metabolism can cause defective arginine metabolism. Defective arginine metabolism can produce hyperarginemia. This metabolic disorder is characterized by deficiency of liver arginase, which normally is responsible for the breakdown of arginine to form urea and ornithine. Since arginase is activated by cobalt and or magnesium cofactors and inactivated completely by ornithine and lysine, these factors also need to be considered during defective metabolism of arginine. So saying it can be from a result of the liver arginase enzyme, there's not enough of it, or we could be low on cobalt, we could be low on, magnesium, manganese, cobalt or manganese, or if we have excessive amounts of ornithine and lysine, which are inactivating it. And that is all of chapter 6. So there's a lot of just straight memorization in this chapter, and I know it takes a decent chunk of time to get through. The best advice I can say is literally take your time going through this video, going through this chapter, pause, do some sort of drawing, do some sort of chart, do something to help you organize and put all of these things where they are, as well as help you organize kind of what the names and stuff of these different conditions and what they're causing. Don't focus too much on like these big names. I would know the first name, like the homogeneous state, the phenylalanine, the tyrosine, the acetoacetate. Know those, like kind of what it's starting and what it's trying to get to and what's wrong with it. But don't try to memorize every single thing. Focus on those. Focus on the amino acids, kind of what they start as, what they become, what they can turn into, and then continue down into make sure you know the enzymes and the names of the conditions and like the most common symptomatology of them. Thank you for joining me for this chapter. So if you have any questions, go ahead and put them down below and let me know if you have any questions. And good luck studying this next chunk of material.